Hello, everybody. I am Alexa Tinsley. I am the Director of Programs here at Jett Foundation. I want to thank you all for being with us here today to learn more about Dyne and the clinical impact of FORCE platform in regards to Duchenne muscular dystrophy. We will go ahead and get started in just another minute. We're going to allow people some time to hop on. But in the meantime, if you are comfortable and able, please go ahead and put your or your child's name in the chat and where you're located. That would be wonderful. And we will be back in just a moment. All right, I will go ahead and get started. Again, I am Alexa Tinsley. I'm the Director of Programs here at Jett Foundation. I want to thank you all for joining us today as we welcome Dyne Therapeutics. We will be talking about the clinical impact of the FORCE platform in regards to Duchenne muscular dystrophy today. So they will be providing an update on the delivered trial of Dyne 251 today. We are so grateful to our partners like Dyne for helping us make all of these webinars possible and bringing knowledge right into your homes. I do want to welcome both Ash Dugar, Chief Medical Affairs Officer for Dyne, and Catherine Beaverson, who is the Vice President of Global Patient Advocacy, onto our webinar today. Before we get started, I do just want to mention a few things. First of all, this webinar is being recorded, so if for any reason you're not able to be on the whole time and you still want the information, if there's anyone that you speak to or know of that was not able to be a part of this webinar today, we will have it up on our YouTube and it will be live on Facebook after the fact as well. We are streaming live to Facebook as well, so if you know of anybody who wants to watch it on that platform, they are able to do so in real time there as well. At the end of the webinar, we will have a live Q&A. So we do have, it's a Q&A, it's a box with a Q&A. For me, it's at the bottom right-hand portion of my screen. We do primarily use that box to answer questions. If you have questions at any point in the presentation, please feel free to put them in there. You can do that with your name, or you can also submit questions anonymously as well. We also do try to monitor the chat to get questions out of there, but there are no guarantees that we will get to those. And also we do try to monitor Facebook as well to see if any questions come through there. Also, another thing that we have is closed captioning. There is closed captioning on the bottom of your screen as well. There's the CC button. If you click on that, you should be able to translate this webinar in real time. And that is all from me. So I will go ahead and hand it over to Ash and Catherine.
Great. Thank you, Alexa. As Alexa said, my name is Ash Dugar. I'm the Chief Medical Affairs Officer here at Dyne Therapeutics. And before we start, I'd like to thank the entire Jett Foundation for the opportunity to present our most recent data release from March at the MDA 2025 annual conference. It is always a privilege to speak to this community, and I want to thank you for your time and attention today. And with me, as always, is our Head of Patient Advocacy, Catherine Beaverson, someone that you know very well and has been with this community for a very, very long time. So, Catherine, if you want to say hi.
Hello and welcome. It's nice to be with the community today. Thanks.
Great. And we're really grateful to be here again. So thank you. Catherine will be can stay on, and of course, will join for the question and answer session at the end of this talk. Next slide, please. I'd like to remind everyone that we will be making forward-looking statements today, and you can refer to our website for more information for all of the details. Next slide, please. So this slide is very important to us. At Dyne, it all starts with our mission: to deliver life-transforming therapies for people living with serious muscle diseases, starting with Duchenne muscular dystrophy. Our mission is woven deeply into our core values and inspires our employees each and every day. Next slide, please. Well, you've heard me talk about Dyne's Force platform, and I'll spend a few minutes on this just as a reminder. The Force platform was developed to overcome limitations of oligonucleotide delivery to muscle for the treatment of rare neuromuscular disorders. One of the biggest challenges that has faced Duchenne and other neuromuscular conditions is the ability to get a therapeutic payload to where it needs to go and to minimize any off-target effects. And that is what the Force platform is designed to do. Now, the platform consists of an antigen binding fragment or an antibody fragment or a Fab, as we call it, that is specific and binds very tightly to something called the transferrin receptor 1 or TFR1 receptor. And I'll talk more about this in a minute. But really, that approach allows us to broadly distribute our disease-modifying payloads to tissues that are relevant to the condition that we're trying to attack. Now, the choice we made was to use part of a full antibody. You can see in this Y-shaped figure in the middle. So we use the top portion, as you can see, that is highlighted in that middle portion. We use this fragment antibody to TFR1 because it doesn't include elements that may lead to greater immune effects, but also the Fab's smaller size makes it easier to get into the muscle cells. Now, the Fab is linked to a payload, and you can see in this burnt orange string the linker, and that connects the Fab to the payload, a PMO in this particular case. And it's this modularity, if you will, of the Force platform that provides us flexibility to be able to use different types of payloads that are rationally selected to target the genetic basis of disease. So, for example, Force can deliver ASO payloads such as exon skipping, PMOs, or phosphorodiamidate morpholino oligomers that need to go to the nucleus of the cell for conditions such as Duchenne muscular dystrophy. Alternatively, we can deliver other materials such as siRNA payloads if we want to attack things in the cytoplasm, such as for conditions like FSHD. But the bottom line is we can deliver to the cellular compartments where they can most effectively target the genetic cause of disease. Next slide, please. So, as I said, the Force platform was designed to overcome the greatest barrier in treatment, which is to be able to efficiently deliver the PMO to where it needs to go with minimizing off-target delivery to other parts of the body. Now, the choice of the Fab targeting TFR1 essentially allows the Force platform to leverage the natural cell biology of our body. TFR1 is highly expressed in the body, including on the surface of skeletal muscle, smooth muscle, and cardiac muscle cells, which enables targeted delivery of the PMO to the tissues that are relevant to Duchenne muscular dystrophy. And our therapeutic agent, our investigational medicine Dyne 251, is designed to leverage TFR1 to deliver exon 51 skipping PMO to the nucleus of the cells in the affected tissues, such as skeletal muscle, including diaphragm and the breathing muscles, cardiac muscle, and smooth muscle to ultimately increase the production of dystrophin. And the goal is to make a shortened functional version of dystrophin. Next slide, please. Now, in a mouse model of Duchenne, the Force platform has demonstrated broad delivery and deep penetration of the PMO to cardiac and skeletal muscle, including the diaphragm. And what you can see on the left-hand part of the slide is that we have very good delivery to tissue that matters. So the diaphragm, again, a respiratory muscle that helps one breathe, the heart, and the quadriceps. And you can see with the Force platform, we're delivering our PMO very robustly to those particular targets. Now, this broad distribution allowed for robust and durable restoration of dystrophin expression and of muscle architecture. And you can see here in these various muscle that we're able to really restore the dystrophin positive fibers within the diaphragm, the heart, and the quadriceps, and there's durability in that effect. Now, in the clinic, we can only explore the ability to deliver PMO to skeletal muscle following administration of Dyne 251, our investigational medicine. In the subsequent slides, I'll show data that support distribution of skeletal muscle and has been translating to early and sustained functional improvement in outcomes in individuals enrolled in our first-in-human clinical trial called DELIVER. Now, distribution to additional muscle types, such as cardiac muscle and diaphragm, are being studied by looking at cardiac and pulmonary endpoints in the trial, but that data will take longer to emerge. Next slide, please. So just very briefly to summarize this initial section, as I said, the FORCE platform was developed to overcome the limitations of delivering antisense oligos like PMOs to muscle in Duchenne muscular dystrophy and other rare neuromuscular disorders. Now, our platform harnesses the natural expression of the TFR1 receptor in muscle, and later on I'll show also in the central nervous system because it does get expressed on the blood-brain barrier. And so this allows for targeted delivery of a rationally selected payload with the goal of addressing the underlying cause of disease. And our preclinical data has demonstrated that by leveraging the TFR1 receptor, the FORCE platform is able to have broad distribution to tissues that are important for conditions like Duchenne muscular dystrophy. And these tissues include the skeletal muscle, including the diaphragm, cardiac, and smooth muscles, as well as CNS. Next slide, please. So I certainly don't have to tell all of you what causes Duchenne muscular dystrophy. It's caused by those mutations. It's caused by mutations in the DMD gene, which result in the inability to make functional dystrophin protein. And PMO-induced exon skipping can restore the open reading frame, which you can see here on the right-hand side of the slide, of the DMD transcript. And by restoring that reading frame, it can be read through by something called a ribosome, and it allows for the translation and production of an internally shortened and functional dystrophin protein. Next slide, please. So when it comes to therapeutic approaches for Duchenne, 3 key considerations are important to keep in mind for any therapeutic agent. Increased dystrophin production, the production of high-quality functional dystrophin, and broad distribution of the exon-skipping PMO to key muscles impacted by the disease, such as skeletal muscle and cardiac muscle. Now, unfortunately, the only currently approved exon 51 skip therapy in some countries, including the US, results in limited increases in dystrophin protein, about 0.3% production of normal dystrophin after 24 weeks of treatment with eteplirsen. Next slide, please. Now, the quality of the dystrophin may also be critical in order to have functional benefit. Here we see a milder clinical phenotype in Becker muscular dystrophy individuals. In this particular cartoon, BMD individuals who carry mutations that produce dystrophin similar to the skip dystrophin results from exons that would also result from exon skipping of similar mutations in people living with Duchenne. So this is a naturally occurring skipping that's occurring to be able to show a milder phenotype. And so when exon skipping occurs in individuals with Duchenne muscular dystrophy, one can expect there to be a big effect on the disease progression. So dystrophin from exon skipping strategies may have functional benefits and lead to improved clinical phenotype. Next slide, please. So in contrast to exon skip produced dystrophin, gene therapy produces a much shorter protein called microdystrophin, which lacks many of the functional domains of full-length dystrophin. Now, there are several different microdystrophin constructs that are either approved, under investigation, or no longer being developed. Now, data on the durability of effect with microdystrophin gene therapy are still emerging, and it remains to be seen if long-term functionality is maintained. Next slide, please. Finally, broad and effective distribution of treatments that generate dystrophin in key muscles is also important for optimal clinical outcomes in Duchenne. And not only is skeletal muscle affected in Duchenne, cardiorespiratory complications are the primary cause of death in children and young adults. And therefore, therapeutic approaches require broad distribution to cardiac and respiratory tissues as well. Moreover, approximately 50% of individuals with Duchenne experience CNS effects, such as neuropsychiatric and neurobehavioral symptoms. Therefore, the ability to cross the blood-brain barrier and deliver to the CNS would be an important attribute for an effective Duchenne muscular dystrophy therapy. Next slide, please. So Dyne 251 is currently being studied in the phase 1/2 DELIVER trial in ambulant and non-ambulant males with Duchenne who are aged 4 to 16 and have mutations amenable to exon 51 skipping therapy. A total of 54 participants have received Dyne 251 across 7 dose and dose regimen cohorts in the placebo-controlled MAD or multiple ascending dose portion of DELIVER, as seen on the left-hand side of the slide in this staircase fashion. I'm pleased to say the MAD portion is now fully enrolled, and please note that the doses that you see here refer to the PMO component of Dyne 251. Now, participants treated during the MAD portion of the study had the opportunity to transition to 20 mg per kg in the open label extension and the long-term extension, OLE and LTE. Participants who were randomized to receive 40 milligrams per kilogram Dyne 251 transitioned to 20 milligrams per kilogram in the placebo-controlled period following review of the safety profile at the 40 mg per kg dose. For other participants, the transition to the 20 mg per kg dose occurred at different time points during the OLE and LTE portions. In this study, the primary endpoints are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints include measures of muscle function like NSAA and SV95C or stride velocity 95th centile. Just a brief word at this moment about SV95C, which I'll talk in more detail about a little bit in a few minutes. This measure has been qualified as a primary endpoint by the European Health Authority and is currently leveraged across multiple global Duchenne trials. While safety data for all participants in the MAD non-registrational cohorts will be shown here, I will focus the efficacy part of the talk on the 10 and 20 milligram per kilogram cohorts. And as DELIVER was designed to be a potentially registrational study, data collected during the MAD portion of the study led to the selection of 20 mg per kg dosed every 4 weeks as the dose and dose regimen for the go-forward registrational cohort in DELIVER. Next slide, please. So on this slide, I show you the registrational expansion cohort. So in this cohort, 32 participants are randomized 3-to-1, meaning 3 active to 1 placebo, to receive 20 mg per kg dosed every 4 weeks of Dyne 251 or placebo. The cohort is now fully enrolled with data planned for late 2025. The primary endpoint is change from baseline in dystrophin protein levels measured by Western blot at 6 months. And a number of additional biomarker and functional endpoints are also being studied, many that you're familiar with, like NSAA and time to rise, as well as SV95C. Next slide, please. So these are the baseline characteristics for DELIVER. And the cohorts are generally similar with a few exceptions. And here I'm showing the 10 and 20 milligram per kilogram cohorts. The 20 mg per kg cohort is on average older with an average worse functioning at baseline, as reflected in the lower NSAA score, as you can see here, and the slower time to rise, the slower 10-meter walk run and stride velocity 95th centile. Therefore, participants in this cohort have more severe disease. Now, based on the NSAA 10-meter walk run and SV95C measures, baseline TTR, or time to rise, may appear as an outlier between the two cohorts. However, participants who could not complete the assessment were excluded from the analysis. And for now, missing times from these participants were not imputed. So that's why the time to rise looks a little bit better in the 20 mg per kg cohort. Next slide, please. So the true goal of therapies in Duchenne should be functional improvement. And we believe Dyne 251 resulted in early and sustained functional improvement versus baseline across multiple functional endpoints, including the North Star Ambulatory Assessment, or NSAA, the time to rise from floor, and 10-meter walk-run time. In the 10 mg per kg cohort, which you can see in the black or the dark line, there is a continued and consistent benefit across these functional measures through 18 months. As mentioned before, upon entering the OLE, participants on 10 mg per kg dose were escalated to 20 mg per kg, with all participants at the higher dose at the month 12 assessment, as indicated by the arrow. Therefore, the sustained effect primarily reflects treatment at the 20 mg per kg regimen dosed every 4 weeks, which again is the selected go-forward dose and deliver. The extension period data for this cohort show the benefit of higher dose and deepening effect on function with longer time on therapy. Now, improvement or stabilization versus baseline are also consistently seen at 6 through 12 months in the 20 mg per kg cohort. And that's in the teal line, as you can see. This is especially notable given that this cohort was more functionally impaired at baseline. So we're really pleased to see this level of functional improvement and or stabilization amongst these different outcomes measures. Next slide, please. So coming back to stride velocity, 95th centile, this was an additional functional assessment that we added to the DELIVER study. It's a digital and objective continuous measure of ambulation in the patient's normal daily settings. It's a real-world outcomes measure. It has been shown to correlate with traditional ambulatory measures, such as the NSAA and the 6-minute walk test. And it's been validated as a primary endpoint in Duchenne clinical studies by the European Health Authority. In fact, SV95C demonstrated greater sensitivity, meaning that it can detect change earlier versus other motor function tests. And very importantly, the proposed minimally clinically important difference in SV95C is 0.1 meters per second. And this is a threshold that corresponds to an MCID for the 6-minute walk test of 30 meters. And so this has been tested rigorously, it's been validated, it's a reliable and reproducible endpoint. And we're very excited about having both the clinic-based measures as well as real-world measures. Next slide, please. So Dyne 251 led to early and sustained functional improvement in SV95C at both the 10 and 20 milligram per kilogram dose. Improvements versus baseline are sustained through month 12 for the 20 mg per kg cohort and through month 18 for the cohort initiated at 10 mg per kg. As with the other functional data assessments, participants in the 10 mg per kg cohort transitioned to 20 mg per kg in that non-uniform way I was describing before, with all at the 20 mg per kg level by month 12. Therefore, the sustained SV95C effect primarily reflects treatment at that 20 mg per kg regimen dosed every 4 weeks. Now, importantly, change from baseline in SV95C following treatment with Dyne 251 achieved the proposed minimal clinically important difference by 6 months, thereby reflecting early and meaningful functional benefit of treatment with Dyne 251. The totality of functional data shows that treatment with Dyne 251 consistently results in early and sustained benefit across multiple motor function assessments and across different levels of baseline function. And this is really important because we do know where you start matters. The baseline function really does matter. Next slide, please. So lastly, the functional benefit with Dyne 251 is driven by the dystrophin produced. This slide shows dystrophin analysis as measured by Western blot for both unadjusted on the left-hand side and adjusted on the right-hand side for muscle content at 6 months. At the 20 milligram per kilogram dose, Dyne 251 resulted in a 3.7% normal dystrophin level. And when we adjusted for muscle content and calculating this with the calculation that's listed here to be transparent, Dyne 251 resulted in approximately an 8.7% dystrophin increase. Therefore, it is the robust production of dystrophin that is driving the functional improvement we observed. Next slide, please. Dyne 251 has demonstrated a continued favorable safety profile with the majority of treatment emergent adverse events, or TEAEs, reported as mild or moderate. Now, we have collected substantial safety data in DELIVER with total doses of administered study drug approaching 1,000 and over 77 patient years of follow-up, with some participants on study drug for nearly 2.5 years. Now, these data also reflect over 500 doses of study drug administered at the registrational 20 mg per kg dose. I want to remind you that there were 3 serious TEAEs potentially related to study drug, which occurred in 2 participants in the 40 mg per kg cohort. Now, these have been previously reported on in detail, and both participants have fully recovered. Since then, there have been no other safety signals of note, and outside of the 2 participants with the potentially related SAEs, no other participants had persistent anemia or thrombocytopenia. And very importantly, there have been no clinically meaningful electrolyte changes, including low magnesium, potassium, or other electrolytes. Next slide, please. So this data set from DELIVER illustrates the promise of the FORCE platform in developing next-generation exon-skipping therapies and supports our ability to expand into other mutations within Duchenne. And this is really an opportunity to leverage the FORCE platform to address those additional mutations. And preclinical programs targeting additional exons, including 53, 45, and 44, are underway. Next slide, please. So I know this is a busy slide. This is just to summarize what I've already presented, and I'll try to hit some highlights. First of all, Dyne 251 is designed to deliver an exon 51 skipping PMO to muscles using the transferrin 1 receptor. And so this receptor-mediated delivery is our solution to being able to drive more of the exon skipping PMO into where it needs to go in a targeted fashion. Now, our preclinical data has shown broad distribution to key muscles, such as cardiac and skeletal muscle, as well as the brain. And DELIVER is a phase 1/2 ongoing trial, and the safety profile is favorable to date. We've also shown robust expression of near full-length dystrophin following treatment with Dyne 251. And very importantly, we are seeing early and sustained benefit of treatment with Dyne 251 that is consistent across a variety of clinical and real-world functional outcomes, including SV95C, NSAA, TTR, and time to rise, and even through 18 months. So functional improvement versus baseline in SV95C, as well as other measures, is absolutely critical for us to be able to illustrate with Dyne 251. And SV95C in particular is a very reliable and reproducible measure of continuous real-world function. And I'm really pleased to reiterate that the registrational cohort for DELIVER is now fully enrolled, and we'll have data planned for later this year. Next slide, please. Great. So one critical aspect that is not often addressed is the multi-system nature of Duchenne. And within that, for a long time, what's gone overlooked in research is brain involvement in Duchenne. Now, of recent, thanks to the amazing work of luminaries such as Professor Montoni and the BIND Consortium and many other efforts in great academic institutions, more research is being done and published in this area. There has been recognition of some core areas of brain involvement identified as particularly affecting individuals living with Duchenne. These are listed here, and some include neurocognitive issues, where people often experience mild to moderate intellectual disabilities with particular difficulties in verbal IQ, working memory, and executive functioning. It also includes academic and learning challenges, especially in reading and arithmetic, significantly impacting school performance. Research has also shown increased risks of anxiety and depression, often observed, and then it's often linked to social difficulties, frustration with physical limitations, and progressive loss of independence. And neuropsychiatric issues are often seen, and this includes higher prevalence of autism spectrum disorder, ADHD, and OCD, likely due to dystrophin-related brain dysfunction affecting the neurons and synapses that are signaling and connecting the different areas of the brain to one another. Next slide, please. So this slide shows that the FORCE platform delivers our exon skipping PMO into parts of the brain and induces exon skipping in the brain of a Duchenne mouse model. Now, given the pervasiveness of neurocognitive manifestations in individuals with Duchenne, as I just mentioned, and knowing that TFR1 is expressed in the brain, we wanted to test if the FORCE platform could deliver our payload to the CNS to potentially address brain symptomatology of Duchenne. So what we used to study this is an HTFR1 MDX mouse model. So this is the traditional MDX mouse model that I think you've heard a lot about, but it expresses the human transferrin receptor. So we can test one of our tool compounds, one of our conjugates, directly in this mouse model. So here in the top row, you can see that 7 days after administration, a single intravenous or IV dose of our tool compound FORCE-M23D, but not unconjugated or naked M23D, or what we call a non-TFR1 targeting negative control conjugate. This led to widespread PMO distribution to the CNS with our conjugate. And this occurred in the prefrontal cortex, in the hippocampus, and the cerebellum, all parts of the brain that are implicated in many of the functional areas that I noted on the previous slide. So we're really pleased to see that this widespread PMO distribution in the CNS also resulted in target engagement as demonstrated by exon 23 skipping seen on the bottom part of the panel in the prefrontal cortex, in the hippocampus, and in the cerebellum. So we're getting our PMO using the TFR1 receptor into the brain, and we're also showing that there's an effect in terms of exon skipping. Next slide, please. So here we see that the effective PMO delivery and exon skipping were maintained 28 days after the administration of a single FORCE-M23D dose in the cortex, cerebellum, and the deep brain. Now, deep brain is a catch-all term for the anatomical region that's likely to contain very small features like the amygdala. Also, this effective PMO delivery and exon skipping to the CNS restored expression of dystrophin as seen in the graph on the very right-hand side of the slide. So through this data, you can see we're getting into the brain, exon skipping is happening, and we're showing production of expression of dystrophin. Next slide, please. Well, what about the impact of the FORCETM23 or tool compound CNS delivery on phenotype? And I'll let you look at this video and I'll talk through it a bit. So it's known that MDX mice exhibit anxiety-induced freezing behavior in response to restraint. So this is a very common mouse behavioral model. And what we saw is that we can actually induce the same phenotype in the mouse model where we crossed it with a human transferrin receptor. So the MDX mouse that expresses the human transferrin receptor. And restraint-induced freezing was assessed by something called scruffing, or where one grabs the mouse by the skin of the neck for 15 to 20 seconds. And this causes the mouse to freeze, as you can see in the top panel labeled vehicle. Now this video is playing on a loop, so the video is moving, as you can see, but on the top panel, the vehicle-treated mouse is not moving. But the force-treated mouse, you can see, is moving. So that bottom video shows what's happening in the presence of force, and it's really exciting to see that we're able to alleviate this anxiety in the mouse. Next slide, please. It's a build. Yep, thank you. So the graph on the right shows that a single dose of force can lead to a complete and durable resolution of up to 2 months post-dose of this anxiety-induced freezing behavior in the HTFR1 MDX mouse. We're really excited to be able to show this preclinical data to you. Next slide, please. So just to recap what we've talked about here, we feel really comfortable, really great that the FORCE platform is able to leverage the natural expression of TFR1 to enable targeted delivery of the exon skipping PMO to where it needs to go to correct the underlying cause of Duchenne. And in our preclinical studies, we show broad distribution to the muscles that matter, like skeletal muscle, diaphragm, as well as cardiac muscle. And in the data that we've generated to date with Dyne 251, our investigational medicine, it has a favorable safety profile with some participants on therapy for 2.5 years. And it has shown early and long-term functional improvement in measures of clinical and real-world outcomes in individuals living with Duchenne. And we believe the FORCE platform has the potential to transform the lives of individuals living with neuromuscular disorders by addressing the totality of symptoms experienced by these individuals, including CNS manifestations. Next slide, please. So I really want to end by thanking the families, the investigators, and the sites that have made this study possible and so many other advancements in Duchenne possible. We know that there are countless sacrifices, the incredible dedication, and what this community, what all of you have done over the years is nothing short of extraordinary, and we're so grateful. Thank you. Next slide, please. And finally, just to thank the Jett Foundation again for the opportunity for us to present to you. And please also reach out to Catherine Beaverson. Her contact information is here, should you have any questions after this talk. And with that, we'll take questions. Thank you.
Thanks, Ash. Again, like Ash said, if there are any questions that you have, please feel free to put them in that Q&A box. I am monitoring Facebook and haven't seen anything come through. But thank you again, Ash, for sharing this with us. It's, very exciting development. So thank you for, sharing them with our community today. If you do have any questions after this presentation, we know that sometimes it takes time to process the information. Sometimes you need to look back over it. Catherine's email is here. If you feel more comfortable reaching out to me to get a question to Catherine and Ash, happy to do so. Alexa@jettfoundation.org. If you are watching this recording, please feel free to also send your questions either to Catherine or myself as well, and we will try to get those answered. But with that, I don't see any questions coming through, which means Ash, you did a great job. Thank you all for being here today. We really appreciate it. We could not do it without all of you. So please look out for our next webinar coming up in May 8th, I believe, on ABLE accounts. And we will see you guys very soon. Thank you all. Have a great day. Bye, guys.